welcome to day 2 of our workshop. So thanks for returning. If you missed yesterday, welcome. And so our goal here at CFDE Pediatric Power Up is to really try to help folks no matter what level they're at, kind of give them a little bit more understanding what types of analysis you can do in the pediatrics world using genomic data that's publicly available through the Common Fund. So here we are on Tuesday, day 2. So yesterday on Monday, we had kind of our introduction to the course. We talked about different types of data sets and different sorts of data that's available. And then today we're going to switch gears. So now we know that what is out there and how to find it. We want to learn, well, what can we do with it? What types of analyses are out there and how are those developed and constructed? And so Sangeeta is going to take us through a great lesson today covering all of these topics. And then looking ahead to the rest of the week, Wednesday and Thursday, we're going to be doing, actually doing the analysis of combining the data we find with some of these methods that we're learning about today. And we're going to try to execute our workflows in Chromatica. And then it all builds to Friday, which is going to be more open-ended, kind of a discussion day, where we try to connect folks with resources to be able to support, your individual project and your individual kind of skill level wherever you may be at. So that's my introduction. And so with that, I'm going to stop sharing my screen and I'm going to pass the proverbial microphone to Sangeeta and I'm going to let her take it away for today.
Thank you, David. Good morning, everyone. Welcome to this day 2 of this Pediatric Power Up. I am Sangeeta Shukla and I work as a bioinformatics scientist at the Children's Hospital of Philadelphia. And I also support this fantastic CFDE project where we're talking about democratizing the data resources available under the CFDE umbrella so that researchers everywhere can more readily have access to the variety of flavors of data. And that's why I'm going to be talking about what we can do with all sorts of data that we have available and easy to access and ready to use. And what we can do on that data is all about conducting all sorts of bioinformatics analysis. So let me go ahead and share my screen and talk about the overall agenda for today's meeting. So all the data that we have access to, what kind of methods can be applied to that and what kind of tools exist out there or we can create to implement any kind of analytical analysis on those data sets. That's what we're going to be talking about. In bioinformatics and the kind of data that we're talking about today, mostly everything begins with next generation sequencing. So I'll start with talking about what technologies have been out there over the years, industry leaders, and what kind of data it generates and how it generates that, and then move on to the kind of analysis techniques that we can apply to those data sets. And, in more recent research events, everything in bioinformatics analysis is now categorized into different flavors where the names of all those different flavors have a suffix omics. And I'm going to talk about all the different flavors of these omics analysis and what they really mean, the kind of data set that they work on, and the kind of interpretation we can get to when we analyze those data sets. And eventually, when we have all that set up and that understanding is developed, I will lead you into talking about the different kinds of bioinformatics pipelines, libraries that can be used to run those kind of analytical questions on the data sets, and how we can manage the entire workflow of having data stored in some place, set up a bioinformatics analytical pipeline to run on that data, and have the conclusion, the results from those pipelines produced, how we can interpret those data. That's what I'm going to be talking about. So let's get started. First, bioinformatics starts with next-gen sequencing. And in this field, in the field of next-gen sequencing technologies, there's a bunch of instruments developed by a few of these world leaders sort of in terms of bioinformatics, next-gen sequencing technology, instrument development. And they have different sort of principles that they work with in the instruments that they build. And these companies, with the instruments that they build and with the principles that they follow, they generate different types of sequencing results. These principles may differ in the sense that some of these instruments may use fluorescent nucleotides to find to tag the nucleotides ATGC or AUGC, and essentially tag those nucleotides by a fluorescent dye, and then that's how they allow the reading of those nucleotides so that we can understand what the sequence of the DNA or RNA may be. Another technology uses ions, H plus ions, to hydrogen ions, to basically understand the chemical composition of the nucleotide that instrument is trying to sequence. Or they can also use waveguides that basically look into what kind of electromagnetic wave each of those nucleotides may emit, and also the electrical current sometimes that they can use to just sort of ping into the nucleotide that they're trying to sequence. And based on all these technologies, all these principles, it's possible to find out maybe like a length of 300 to 400 base pairs of nucleotides in one read. Or if the instrument is more evolved, it can read up to a few thousands or hundreds of thousands of base pairs at a time. That's called the read length of the nucleotide. And what happens is, in this process, the same nucleotide is broken down and amplified and read multiple times. And when it's read multiple times, and the longer it is, that the word for throughput is basically just meant to say that this is how quickly the instrument that you use, the technology that you use, can produce a read that is more in the format that we can use for downstream analysis. So the longer that read is, the lower the throughput. That's how, if the read is a lot longer, then it's not as quick or not as big as you think, because you'd want to accommodate for the instrument would have to accommodate for the time to read that sequence, and before that it'd have to run all sorts of chemical reactions on that nucleotide sequence. And then based on the kind of instrument you use and the read length that it produces, the accuracy of the overall sequence also may vary across these different technologies. But overall, all of this data is meant to be used for downstream analysis. Now, this data could be at the genome level, where it's a whole genome sequence that you're trying to extract from a sample, or RNA sequencing, or some microbial genomes that you're trying to extract from sequencing, and then perform analysis downstream. And those questions on what kind of analysis we may perform on this data, that we're going to be talking about in just a little bit. One important factor we also want to consider is that over the years, the cost of running these kind of sequencing experiments has definitely become more, well, not exactly affordable, but it has gone cheaper, from being very expensive to slightly expensive. And yet, although this has evolved, the cost for running these sort of sequencing experiments is still moderate enough. However, there are also sometimes some of these smaller companies send coupons out there to just, if you wanted to, you could get your own genome sequenced if you're interested in. And sometimes that's a good idea as well. But yeah, that's the kind of introduction to next-gen sequencing technologies and the evolution of these technologies over the years. Because, if you're familiar, even the Human Genome Project, that concluded like 20 years ago. So it's been like 2 decades since then. So it's understandable how vast the evolution may have been for these technologies, and also the costs for these technologies over the years. So these, once you have that next-gen sequencing data, what do you do with it? You have done the sequencing. What happens next? And in general, typically all these NGS analyses steps, they follow a similar pipeline. You start with sequencing, you do some pre-processing, some quality control. Essentially, that means you want to inspect whether all the reads were captured appropriately, whether if there's any noise, you want to find ways to minimize that noise and make that data more usable, and then perform some adapter trimming, because as I said earlier, when you want to perform these sequencing experiments, you have to amplify the sample first of all. And for amplification, you also need an adapter to which the polymerases can link to, and then perform that amplification. And then eventually, when you have the sequencing data available, you want to pre-process that to remove those adapters that you had attached, and then also verify what kind of quality data you have now. The data has to be read more efficiently. There has to be less noise. The adapters have to be trimmed appropriately so that the downstream analysis does not have any interferences. So that sort of quality filtering, once you have done that, the next step would be to understand how to bring that genome back together. Because as I said, for the amplification, you have already broken down the sample into multiple pieces. And that's why the sequencing reads that you were able to get by running the sequencing experiments, they are now in different fragments. So you have to find a way to put those fragments back together. Now, if you knew what the genome originally was like, if there was a reference genome, you could perform like an alignment to the reference genome for all these fragments together, so that you know how those fragments come back together and align to the reference genome. Or if in some cases the reference genome is not available, then there are methods to basically perform what's called a de novo, from scratch alignment, mapping, de novo assembly of the genome that you've just broken down and amplified and sequenced. There are different algorithms for that as well, that we can, if we have some more time at the end, I can give you an insight into that as well. And once you have that kind of alignment or mapping or assembly done, the next step is to understand what kind of variation it has from the reference genome. Because this reference genome was developed and has evolved over the years, but depending on the kind of sample you use, the kind of it could be like a healthy sample or a non-healthy sample, or it could be from a different kind of phylogeny, somewhere a different phylogeny it could have been extracted from. And that's why a variant calling will help identify the different kind of evolutionary artifacts that might have been introduced into your sample. These artifacts could have been single nucleotide polymorphisms, or insertions and deletions, or the overall structural variation. If you were doing a genome analysis and there were the DNA sequencing was structurally different from the reference genome, or it has evolved a little bit. And there are various tools out there to be able to conduct this kind of variant calling. And the most widely used of those tools are GATK or SAM tools or VARSCAN. And once you have those variant calling variants understood and called and captured into what's called a VCF file, the next step, depending on the kind of data that you're working with, is quantification. If you're working with RNA-seq data or some protein data, quantification would essentially mean that you capture the levels of expression of genes or the abundance of proteins into files, so that you can understand and compare how, given the kind of condition that you extracted your data in, the biological condition, what is the level of expression of certain gene in a healthy or non-healthy sample that you're working with. And there are a lot of tools out there for doing this kind of analysis as well. Feature counts of salmon, or like a more recent version of, like a evolved version of salmon is 11. So there are these quantification tools out there also to understand how to capture the data from like the basic sequencing kind of raw data to pre-processing to variant calling to quantification. And this cascade of the data makes it more it adds a level of complexity to the kind of information you can extract from running these sort of tools on the sample that you are working with. And essentially, when you do all this, the goal is to be able to annotate and analyze what sort of functions biologically this data may be involved in. And for those questions, for those annotation and functional analysis, when it comes to understanding what sort of if you were to run some experiments like analytical experiments, run some libraries to run that variant calling or quantification, how would you understand that, okay, now the resulting data, I have it in a certain file, but what am I going to do with it? How do I understand what is written, what is captured in those files? And to be able to do that, to be able to understand that analysis, we would need to do some integration of all the different files and understand what they have captured in that by visualizing that data. And once you visualize, it becomes easier to interpret what all the experiments that we have run, they really mean in terms of their biological manifestation, essentially, what it means, and how we're going to understand and share that data, report that data to the scientific community. So that's like the basic steps of next-gen sequencing data analysis. You start with sequencing, you pre-process the data and prepare it for the next step of alignment or mapping or assembly. And then you run a variant calling on that data and appropriately run quantification tools on that data to finally be able to annotate and analyze that data so that you can share it with the scientific community. Okay, so once you have that data, and you want to run an analysis, the kind of analysis is also categorized into different omics analysis, depending on the kind of input data that you use. And it can be made more complex in terms of, in terms of using not just the input data, but also investigating the kind of biological manifestation that it leads to. And that's why these categories, if you were to start with just the central dogma kind of sequence, like you want to work with the genomics data first, and you could just run genomics analysis, the genome, the DNA analysis, and then you could look into transcriptomics, which essentially is about understanding and carrying out analysis on the output of transcription, which is RNA. We have more questions. No problem, Bijon. It's all good. Thank you, Bijon. Thank you, Hang Chang. Yes, so as I was saying, yes, transcriptomics. Transcriptomics, as you can see from the name, it involves working on the data that we can derive from transcription. And from that, we can also move on to proteomics, which is essentially understanding how the protein, which is of course the result of translation of the transcribed output. So when we have those proteomics data, when we have that protein information, we can also work on that protein data to understand the kind of biological manifestation of that, and that's called proteomics. From that, we can also look into other more evolved kind of categories, metabolomics and epigenomics and metagenomics. But don't worry about this slide, because I'll be talking about these in more detail in the next slides. So let's move on to that. Genomics, as I was saying, it does involve working with the DNA data, but how do you exactly do that? What is the general, steps of working with genomics analysis? What do you start with? What kind of analytical knowledge can you gain from it, and what kind of interpretation can you gather out of the genomics analysis? Essentially, the genomics data is all about genomics analysis, is all about getting your sequencing data, running like a quality control of that, and then reading using the reads to and aligning them to a reference genome, and variant calling, which is essentially what we talked about earlier, like the basis of all of NGS analysis pipeline. And once you have that, what you can do with this data is run what is known as like a genome-wide association studies, or just annotating the variants across the same species in different health states, or across species on genes which have the same function, or understand the differences in the copy number variations, because sometimes in different individuals in different ethnicities it's possible that the same gene may have like different copy number variations, or also the structurally these variants could be structurally different, and use that kind of data to sort of run a comparative genomics analysis and understand how across species or within species across different health conditions or ethnicities, or races, the same kind of genome may have evolved, have varied over the years. And we could use this analysis to essentially understand some phenotypic traits or diseases, and use the knowledge of those genomic regions by annotating them to understand what kind of diseases may those genes or genomic regions may have been involved in, and then identify candidate genes for further study or sort of disease investigation or disease treatment. Do we have more questions? Sorry. You want me to be more specific, for example, which tools are available in Quadica? Yes, absolutely, yes. So that's going to happen tomorrow. We're going to be talking about the different tools and pipelines that we have available pre-existing in Quadica, and also find out ways to see how we can create custom analytical pipelines that we may have been working on our end, on our lab machines, and how to take them on Quadica so that they could be used more collaboratively. That's what we're going to be talking about. Yes, we will be talking about these tools available in Quadica tomorrow. I see that a lot of you have already been working on more advanced topics already, and it's possible that a lot of you know some of these omics flavors already. So that's great. Please do chime in whenever you find something interesting and you have a tidbit to share. From proteomics, let's move on to the next topic, which is the next version, the next level of biological manifestation, metabolomics, and then transcriptomics. From genomics, let's move on to transcriptomics, where transcriptomics is about extracting the RNA-seq data and then running through the same steps of aligning to the reference genome, and then quantification of that gene expression levels, and using that knowledge to analyze and compare the differential gene expression across various genes under various conditions in the same or different organisms, and using that to essentially understand the kind of biological processes or conditions that are triggered by these varying differential expressions. So this can essentially be used in answering questions where we want to understand regulatory network, like at an infant level, what kind of regulation happens on certain gene. If you grow up in adult stage, what kind of regulatory level can happen for a certain gene, and do genes work in tandem with each other? Or is there like a co-expression that we see in some genes? Those kind of questions. That is what we can answer when we run transcriptomics analysis. The next level of complication that we can add to our analysis is proteomics. So we can this can be a little more versatile than transcriptomics, where we work with the proteomic data and run experiments like the mass spectrometry on that protein to essentially read what those amino acids may be, what the sequence of that protein may be. You want some Python or R packages that will run the same analysis without Quadica servers. Oh, great. Bijan is working on some gene expression analysis. Great. So yes, Haroon, that is the next question. We're going to be talking about some of these Python or R packages that will run that can be used for proteomics or genomics analysis. And yes, Quadica servers are not essentially just the sources of R or Python packages, but they are just like the packaged environment where we could use these R or Python packages to implement these analysis. But yes, for these analysis we're going to be talking about later, I'm going to be talking about the kind of biological questions that we can answer by running these sort of omics analysis, and what kind of packages can be used, and whether they are Python or R as well for running these kind of analysis. But right now, as a precursor to that discussion, I will just briefly go through the different kinds of proteomics analysis as well. Absolutely, bioconductor, and that's of course biopython as well, which have a variety of these Python and R packages that you're possibly looking for. Hope that helps. Okay, from proteomics, we move on to metabolomics, which is essentially understanding how the proteins that were created as a result of translation, how they are being metabolized in the biological composition in the organism, be it human or non-human, whatever organism that may be, or plants. And for metabolomics, it again requires mass spectrometry or NMR data to understand what kind of amino acid sequences we're talking about, and then identifying these what metabolites these constitute toward. And based on that metabolite and the abundance of that metabolite in the sample, we can essentially identify and associate these metabolites and their abundance with the biological processes or conditions, and find inferences of the metabolic pathways, where we can understand, we can see what kind of changes we have seen from like the genomic level to the transcriptomics level to the proteomic level, and then finally understanding how the entire metabolic pathway works. And this can lead to basically discovery of new biomarkers, which can be used for disease treatments or disease diagnosis, or at least just prognosis, eventually finding like new personalized treatments for various diseases. All right, so after metabolomics, the next level of more complexity that can be added to the data is epigenomics, where we talk about how external factors may have impact on the genomic composition. External factors like maybe radiation or climate, or the food that we eat, or the lifestyle overall, and how that kind of external factors can trigger some changes into the genomic composition. And the study of that impact is called epigenomics. And for epigenomics, the overall data is essentially the methylation data in the DNA, the kind of methylation that occurs. That is what we are looking at when we want to conduct epigenomics analysis. Once that DNA methylation is the DNA methylation data is extracted, we can again run alignment algorithms on that, and then normalization for the expression levels, and then run like a peak calling to identify what kind of histone modifications have happened structurally on that genomic data, and then run analyses such as identification of those histones, differential, of course, differential methylation analysis, and then identification of the regulatory elements when we want to identify the promoters or enhancers that lead to the expression higher or lower of the sample that we're looking into. And we can also look into motifs that may exist. Motifs are essentially just like the signature kind of genomic sequences or proteomic sequences that may be found as a result of some kind of external trigger um, in a sample, and use this sort of data to understand how the external factors may lead to phenotypic traits, and what kind of markers we can identify to see how external factors may cause diseases in different organisms or plants. And the next step of this is also understanding the metagenomics. Metagenomics is all about also understanding the impact of other genomics, other genomes also on the sample, which includes like the environmental um, microbial communities which may have impact on the sample that we're looking into. And again, for this, the pre-processing would be the same as getting the sequence data, doing some quality control experiments on that, and then based on the sequencing data, we also want to. Yes, we have one more question in the chat. Yes, so these data may come from the same experimental designs. For example, for metagenomics, if we're working with some kind of studies of some sort of virus, let's say COVID virus, let's say, for example, the COVID virus, we're working on the people who were impacted or had showed morbidity due to the COVID virus. The kind of analysis that we run on that would technically fall into the metagenomics category, because then we're looking at how an external genome was impacting the patients who were impacted by the COVID virus. So they would come from the same experimental design, but they would still have to be studied as the host would have been impacted by the external genome, right? So we want to study what kind of changes, metabolic changes or genomic changes have happened in the host because of the, yeah, something like that, host pathogen interaction. Yes, essentially. But it doesn't have to be just a pathogen. It doesn't have to have a negative impact on the host. It could also be just like a symbiotic impact, which is why, those kind of, there are those like um, uh, what is the word, symbiotic relationships for when you want to consume. Again, sorry, my the chat window is closed sometimes. Yeah. So exactly, so we can have symbiotic pathogens, symbiotic interactions as well with those hosts. That's what metagenomics is all about. And this would essentially start with, again, capturing that data, finding the sequence of that data, and identifying what external microbial or viral taxa may have impacted the host at this point, and what sorts of genes or pathways have been impacted by that. Yes, gene environment interaction. Yes, absolutely. That's where this is, gene environment interaction. Yes. Metagenomics is all about finding the combination, understanding how the relationship between the host or a pathogen in the given environment may work in tandem to generate what kind of manifestation it may be biologically, good or bad, both. And then once you have that understanding, we can also take it to the next level called phenomics, and then understanding systems biology and network modeling. Phenomics is all about investigating and understanding the phenotypic differences that the underlying data, underlying metagenomics or metabolomics data or proteomic investigation may lead to, because all that data is at the genome level or at the proteome level to just understand the differences in the protein abundance or the gene expression levels. So that's at the molecular level, but phenomics is all about understanding the phenotypic manifestation of those investigations. Again, here it would essentially involve a lot of data cleansing and normalization, but it would also involve what kind of phenotypic features we want to work toward investigation, and then using some sort of statistical tests to analyze that phenotypic data. And this is where, a lot of these modern bioinformatics and machine learning analysis that we hear about, this is where they can be put to use more commonly. Of course, there are other reasons why we'd want to use machine learning or advanced algorithmic analysis, algorithmic methods to investigate biological data as well. But this is one of the more widely used ones, because this is the kind of the investigation where we look at phenotypic differences, and that's why running like clinical like a drug, how a drug may impact biological environment, those kind of data may look for phenotypic investigation as well, because we want to look at how a certain biological pathway was impacted by maybe a drug or an external factor or climate change or something like that. So this is based on essentially the genotypic or environmental factors leading to phenotypic changes. And a more broad category of bioinformatics or computational biology analysis is also systems biology and network modeling, where the goal is to understand the subcellular structures and how they work with each other. In case of like a systems biology based investigation, it's possible to model a biological, a biophysical structure like a cell or a mitochondria and capture the chemical reactions that happen inside it. We can extract the size and the locations of the substructures like ATPase or different sorts of or just the membrane size, or for example in mitochondria, how the crystal works and the size of ATPases and the locations of ATPases and the quantification of ATPases that are present on, let's say, mitochondrial cristae, and use that sort of data of a subcellular structure to simulate the biological, biophysical, and chemical reactions that happen in a smaller space, and use that to understand the changes in the volumes of the metabolites that are part of those reactions. So that is the another example of like a different flavor of bioinformatics computational biology analysis as well. And on the other hand, once we have a lot of information about different genes or different proteins or different metabolites, or understand some phenotypic manifestations of interferences in those genes or proteins or metabolites, what we can also construct is like a network, a regulatory net, a biological network, and represent relationships or interactions between the biological entities that we have knowledge about. And these networks could be represented as graphs, where nodes represent the entities and the edges represent the relationships or interactions between them, and use that diagram, use that graph to essentially understand and identify how gene regulation happens in different cellular processes, what kind of signal transduction mechanisms are at play, to sort of understand how the crosstalk between different pathways may happen, and how different diseases may be linked to a certain gene or a certain set of genes, and use that kind of network information for a more focused biomarker discovery, or also use that kind of information eventually to understand and create more personalized treatments for diseases, essentially in the long run. So of course this relies on a lot of knowledge brought together, but then we have all these other methods that we talked about earlier that will bring all of that data together. And once we have the data, it just makes sense to find out how they all come together in like a network. Yes, now we were talking about, Harun was looking for different kind of R and Python packages that we could use for running those sort of analysis. So yes, here is Harun's answer. Depending on the kind of data that we're looking to work with, whether it's genomic data or transcriptomic data or some proteomic data, there are different sorts of bioinformatics pipelines, which are essentially, they're not exactly preset, but they follow a certain set of rules to start with, and to essentially run through an entire sequence of steps to run that analysis. And once you have that analysis, the kind of visualization that we can get out of that investigation can lead to identifying a certain type of data, certain type of knowledge that we can extract from that investigation. And these bioinformatics pipelines can be categorized into like a differential expression analysis, where we work with either the genomic data or the transcriptomic data, and understand the conditions, different conditions which cause those genes or transcripts to be expressed differentially given the conditions. Once we have that information, once we understand that, once we have the knowledge of the differentially expressed genes, we can also conduct what is called a functional enrichment analysis, which allows us to, okay, there's more in the chat. Yes, absolutely. Yes, that's right. Data from qPCR or microarray. We can also use neural network for the kind of network analysis that we were looking at earlier. Yes, and PCA can also be used for creating that kind of dimensionality reduction. Yes, absolutely. I'm glad that you're all enjoying these topics and have things to contribute to. Yeah, I'm excited. Thank you for all the input. Yes. All right. So the bioinformatics pipelines. Yeah. So once we have the differentially expressed genes, the next step would be to run a functional enrichment analysis on that, so that we understand the different biological functions or pathways these genes are involved in, or may have been involved to impact those biological functions. Other analysis would be like an alternative splicing analysis, where we want to understand from the transcription level or the translation level, the different kind of alternative splicing events that may happen, which generate different isoforms of the transcription output or the translation output, and then quantify that to see how that impacts a certain biological function or a certain biological pathway. And when we have that understanding, we can also work with some of the proteomic data. For example, in case of ChIP-seq data, which is essentially chromatin immunofluorescence kind of protein sequencing, and then use that data to understand how the enrichment of a certain genomic region happens with the protein being in the close vicinity, and how that impacts the overall structure of the genome and the proteome, and what that leads to downstream in terms of the biological pathway and how it manifests into an impact like a phenotypic feature, and use that knowledge later again to understand the entire pathway of how a biological pathway may be impacted by the genes or the proteins essentially. And the next level would be just to bring a group of these kind of enriched data sets together to also investigate if there is a possibility of some sort of coordinated changes happening, which lead to some phenotypic changes. And again, this leads to an overall bigger understanding of like a network where we can understand the different protein interactions that can happen, and how they lead to some sort of metabolic or phenotypic manifestation. So these are the different kind of analysis by like actual pipelines that can be run, and these would be a combination of this analysis would have to be essentially a combination of a bunch of R or Python packages. For example, for differential expression analysis, you'd start with like DEC2, which is an R bioconductor package. And there's also, if you were to conduct a single cell analysis, a single cell RNA-seq analysis, then you'd probably want to use a different, more specific R package, like a single cell experiment or something. And similarly, there are these other R and Python packages that can be used in conjunction with the rest of the analysis to conduct an entire end-to-end investigation pipeline. Once you have an understanding of the kind of investigation you want to perform, with that understanding, you can start with running an actual experiment, investigative experiment. And for that, you could either have some data that's available to you from your lab, or you could use some of the public resources out there. And typically over the years, a lot of resources have been built for storing and sharing biological bioinformatics data. And these are mostly widely known, but a general list of that for genomic data would be some of the NCBI databases or some transcriptomic data. If you're looking for that, you could look into GEO or SRA, which is Sequence Retrieval Archive. Or if you were looking at the metabolomic data, you also have the HMDB, which is slightly older homology modeling database. HMDB is homology modeling database, and there's a more recent version of that, which is called the metabolomics workbench. Or for the entire pathway interaction data, there's KEG, which is Kyoto Encyclopedia of Genes and Genomes, and Reactome, something like that. And if you're looking at structural data for proteins, PDB has been the most widely used data repository. And for epigenomic data, more recently, ENCODE has been widely used. And for disease-specific data, there's of course TCGA for a lot of cancer data, and ClinVar also for a lot of data. But this is essentially to just share the variety of data that can be captured in different databases. And under this CFDE umbrella, the CFDE workbench, that's right, PDB is for the structural proteomic structural data. PDB is essentially protein data bank. So yes, it has a lot of structural data for proteins. And for all sorts of these flavors of data, under the CFD umbrella, the different DCCs that David was talking about, they have also captured a lot of such data from different organisms and different disease states, and pediatric and adult data as well. And all of that data is also available on that CFDE workbench that David was talking about yesterday. And with that data, CFDE and others, that individually or in combination, that can be used to investigate the kind of biological bioinformatics pipelines that we talked about earlier. But to be able to successfully do that, to be able to successfully run an end-to-end investigation, you would need to think about a lot of aspects. You would have a lot of data that you're working with. And these data files aren't small, they are huge. So once you have like huge data files, you need enough and properly organized setup to store all that data. It could be your on-premise machines or public databases. But public databases, absolutely, Bijon. You are right. That's what we're talking about here. And this is like a segue for me to give you like a general idea of what an entire end-to-end bioinformatics analysis workflow will call for, and then how Kavatica comes into place to enable users, researchers to use a same environment, one common environment, to have all of these workflow requirements all put into one place, also making it collaborative so that you could work with people within your lab or people with other labs, or anywhere across the world that you could share your work with, or have collaborative ways of having everybody work on the same setup as well. So yes, as I was saying, when you have huge data files to work with, not just in terms of the size, but in terms of the number as well, you would need a setup to store all that data. You could have your on-premise machines for data storage, or you could put them on cloud services. And cloud services, of course, that also incurs some cost. But once you have those cloud storage set up, you also have to be able to interactively retrieve those data that you've stored on premise machines or on cloud services. And once you have that set up and you are able to retrieve them, the goal is to be able to link them to a certain bioinformatics workflow that you're trying to process, so that you could use that data and run some data cleansing and harmonization steps on them, and then run through the analytical pipeline of interest that you're looking to work toward, and use that pipeline to extract an output which you can then visualize. And essentially this is all just like a entire software development kind of process. It's what in the software terms, in the IT terms, called like a software development life cycle, where you have to have the data, you have to have like a pipeline, you have to have some QC steps on the software that you run, and then you have to be able to have the visualization that you need at the end of it, so that you can have the interpretation from that workflow processing that you've performed. That's like the entire software development life cycle. And to be able to do that, it's not enough that you absolutely development and OBs, yes, absolutely. And it's not enough that you only know how to store the data, or that you don't only know how to run the software development. You'd also need to understand how to use the computational resources, basically how to enable a higher amount of resources to be used at a certain time, and then clean up those computational resources so that newer data can be processed or stored on that data. And then you also have to understand the difference between when you need more resources for storage versus more computational resources for data processing. So that kind of management of the computational resources also needs to be understood when you run such an experiment. So it's not enough that you just know, the formats, the different data formats that these biological data files may be stored in, or just the biological pipelines, bioinformatics pipelines that you may need to use to run on certain data. That is not enough to be able to run an end-to-end experiment. You need a lot of these computational resources. And also, when you work collaboratively, when you want to work on a project that is going to run for a year or two or more, and you're going to have different versions of your software evolving over time, working with initial data or a newer version of the data, or a more comprehensive investigation that you're performing on your data. When your software is evolving, you would also need something called like a software management, code management repository that can handle this kind of evolution. And it can allow for the users, the collaborative users, to work with this code repository, a management tool, where you can introduce newer versions and allow your code repository to also expand, but also allow for bugs if they are found to be fixed, and then allow for that same code repository to maybe run the entire set of software pipeline individual steps to work on the newer data. So let's say step one, I had 5 scripts that I was running on 10 files, but in iteration 2, I found a bug, and out of the 5 scripts that I was running, 1 script needed an update. But now that I've updated that script, I have to use the same 5 scripts and run them on the same 10 data files that I had. But in the newer version, I might want to introduce a newer analysis and use the 6th script as well. And then I'd need to run the same 6 scripts on that same 10 data files again. Let's say in the newer version, I have another 100 data files now, and I have a total of 110 data files. And then I want to work on the analysis of the 110 data files. Then I'd need enough computational resources to be able to store those 110 data files, to be able to run my 6 scripts through processing the 110 data files. So I need enough storage and processing power to analyze all that. And then again, I would have to be able to visualize the results from processing those 110 data files, so that I could make newer interpretation. So this kind of CI/CD continuous implementation, continuous deployment kind of setup, that's what you also need. So when you have this kind of setup, essentially what you're doing is you are making sure that you have enough for your data storage. You have to know what the bioinformatics workflow is going to be like, what kind of analytical pipeline you are using and using to implement on how many data files, and what kind of computational resources you need to essentially make sure that the process of data storage and data processing is efficient. And you need to also account for the code repository, because the time that you would work on this data set for may not be just like a short week or a month or two. It could run for like a 3 or 5 year duration. Hopefully you have funding for all of that. But yeah, the point I'm trying to make is that this process is not going to be only limited to a certain individual user or developer. You might also have to work with a group of people within your own lab or with other collaborators, and that's why you want to be able to make this process of uh, having to store the data, running the bioinformatics pipeline, and code collaborative. And when you make things collaborative, it's possible that the systems that your collaborators work with, that may be different. And that's why some of the computational dependencies or the software dependencies that you may have on your machines, your collaborators may or may not have them. And that's where the concept of software containerization comes in. What I'm trying to get at is that a bioinformatician may be apt at understanding bioinformatics data, but in the modern scenario, in the modern world, in the modern world of bioinformatics research, it is also absolutely important to understand the entire software development lifecycle and the DevOps kind of a setup to be able to run a collaborative, time-dependent kind of analysis that you may work toward. Absolutely, Bijon. Which is why the software that we're talking about, the platform that we're talking about, Kavatica, is helping the users to get all of this sort of limitations dealt with without having to learn them individually. So if I was a Kavatica user, which I am, I wouldn't need to individually worry about where I'm going to store my data. If I have a bioinformatics analysis pipeline, how I'm going to run all the data through that pipeline, what kind of computational resources do I need? Can I share my work with my collaborators and get their feedback, get their input? How do I convert all of my work into like a publishable format? How do I work with the kind of software evolution that keeps happening, even if I were using like a simple library like let's say DEC2, simplest of, let's start with that. But DEC2 also has versions. How am I going to keep up with the different sort of software versions that my bioinformatics workflow is using? I'd have to know all of that. But with a tool like Kavatica, a user would be able to have their data stored on AWS or Google, AWS buckets or Google Cloud storage setup, have the bioinformatics workflow available to them in the form of pre-built applications on Kavatica, and you would also have the option of having your custom pipeline be it in any language, R, Python, C Sharp, C++, whatever it is, converted into a CWL file, and then it would again work as an app for you, which you could also share with your collaborators. The computational resources that you would need to work through your data files to store them and process them, they will also be uh, handled by the software, by the tool itself, by the platform itself. And you would also be able to invite your collaborators to your project, have them you could give them like an admin privilege, or just let them execute the data, or let them just read through the result files that you have without having to worry about the entire setup being replicated in a different location as well. And R also allows for these pipelines to work with the newer versions of the packages like R or Python. So you don't have to worry so much on your own about working with the evolution of the libraries that you use. Or if you had already set up a code repo, you just link that code repo with the Docker Hub to your CWL file on Kavatica and use that without, again, having to track the version of everything, to iteratively work on the data files through the bioinformatics workflow, and also allow for your collaborators to work with that. So that's essentially like an introduction to what Kavatica offers. But it's not just that it offers that. We'd have to talk about what kind of um, storage costs it will incur. And that's why we were talking about yesterday when David introduced all of you all to, okay, so the rest of it is a conversation. So I'm going to stop sharing my screen. So when you have all that data set up, when you have all that computational resources allocated to you, and you know what kind of bioinformatics analytical workflow you're processing, sure, Bijan. When you have that, it's the next step is to be able to actually run that analysis. Is the app building expected to be on the user? No, not entirely. Some of these apps exist pre-built on Kavatica, so you could just plug and play kind of things. You could just open an app, for example, the app that David is going to demonstrate tomorrow. You could just plug in your data files, click like a run button, have the data set processed, and have a visualization for you prepared. So that helps the task of software development, computational resource management, code management dealt with entirely, and you could focus on just your bioinformatics investigation. Does that help, Uma?
Hey, what about if you have you're thinking about a new app which is not there and you want to build one?
Absolutely. So let's say that you have like a bunch of scripts written at your end, each of which is working on different steps, starting from pre-processing to some kind of pipeline analysis to the next being like a visualization, and then maybe there's a shell script which is calling all of this, like like a workflow management kind of script that you have at your end. Let's say you have that, and you know what kind of data set you want to work with, but you just don't have the compute storage. You just don't have the data storage set up at your end, or you want to work with other controlled or public data sets that you work with your collaborators. So if you have that, you could just take all of your scripts and wrap them into CWL, which is command line workflow language, common workflow language, and upload it, like deploy it on Kavatica, and then make it collaborative. You could share it with the members of your own team or with your collaborators, and also help them use your app essentially to perform their analysis on their data sets or your data sets. That's what you can do. So you don't have to build an app always, but if you wanted to for some custom analysis, you could do that. And that's
So
what
are we going to be, is the CWL going to be part of this training? I mean, I have not worked with that language before. If I have a script and I have a workflow and I want to convert it into that, is there any help?
Yes,
Expected
absolutely.
here? Okay.
Yes, the short answer is yes. On Thursday, I will show you how to use your scripts and wrap them into an app. That's what we're going to be doing on Thursday. But yes, if you had your scripts and you wanted to wrap into an app, you don't have to know CWL entirely. I'm going to show you like a cheat way to still create an app when you don't know everything that the Kavatica developers may know of.
Okay, that's good to know.
Absolutely.
Thank you.
No problem. Yes, so essentially what I was talking about is how to worry about less and get more done. When it comes to bioinformatics analysis, you could just focus on your own data and the kind of pipeline that you want to implement to process your data set, and have the visualization ready to interpret, and not worry about the storage needs or the compute power or the code sharing or the evolution of the libraries that you work with. All of that can be taken care of on one collaborative environment. And that's what we're going to be talking about tomorrow when David demonstrates how to use an existing pipeline on Kavatica. And the day after, when I talk about how to build a pipeline from scratch, either with your own data or some data that you download from public repositories, or if you were to run into some issues, how would you tackle that? All of that. That's what's happening tomorrow and the day after. So for now, I think I will run through all of my content really fast. So I'm very happy to answer any of the questions you have. If you have questions about what sort of data you are working with and what is next for your data analysis needs and how Kavatika can be used for that, let us know. Tell me something, how many of you currently use cloud resources for your data storage or data processing needs? And how familiar are you all with cloud computing?
On
And
my
rating
part,
are fine.
I mean, I don't use cloud computing at all. We have servers, so we are happy with that. So that's how we do it. Kavatika was the first cloud platform which I tried, I would say. And I would like to know more on that because I am mostly a command line user. So I am more comfortable on the command line than the app basis of things. But then Kavatika helps you to work with the data sets which are up there. So I'm interested
Definitely.
to know
And
more. Yeah.
you have to collaborate with others for your
Not exactly at this point, but then it would be in the future, definitely.
Okay. And all of the other people, how many of you are comfortable with cloud computing? You can raise your hand on Zoom. No hand
I
raised?
was just going to quickly say that I'm familiar with the concept. I haven't probably spent much time doing cloud computing bioinformatics work, but yes, my institution is having a cluster where we can work from. But yeah, I mean I'm pretty much novice into bioinformatics, but um I haven't spent much time doing. I have just been working remotely, sometimes trying to use other resources that exist online rather than having to use command line. So yeah, I'm in between the two worlds again because I'm novice. Yeah.
So because because you work with a cluster, you might also need to manage all the software issues and installations and everything at your end.
I think there is a facility that is responsible for like managing the architecture and stuff like that. I mean, I was extensively using it for like the genomic side of things. Now I'm transitioning more into the transcriptomics, and I think that it's just best for me at this time to leverage other existing cloud-based apps to do the transcriptomics analysis instead of like using command time now. That's what I just think. Again, people may have different thoughts around that, but again, that's what I'm doing now instead of. And I guess the Kavatika, I think it's an interesting platform. I haven't used it. I've been trying to use Galaxy, and again, I think these are great platforms.
Absolutely, yes. Galaxy was one of the most interesting platforms a few years ago when bioinformatics analysis started getting, when you had to use more resources and not everybody had those resources. But from what I know, it doesn't allow for data storage and for long-term data your storage and compute options. So it just allows you to temporarily plug and play kind of options for running analysis, right?
Correct.
Yeah, as compared to that, this is Kavatika essentially is like the one-stop shop for everything. You could be working at a hospital on like multiple data sets that are proprietary for the hospital, or you could work with public data that you could extract from any of the public repositories out there, and you could bring them over to Kavatika and run through a workflow pipeline that you may have built, or like a custom pipeline that you may have built, or the pre-existing pre-built apps that are available on Kavatika, and you could run those analysis, have the visualizations prepared, ready to be written about in a publishable format as well. So it just essentially allows for bioinformatics researchers to get more productive without having to worry about the other aspects of running an analysis in the modern world, where you have a lot more data files that you may need to process even to generate like a small experimental conclusion. And sometimes, universities or companies may have a lot of that setup already done, but sometimes you may not. You may be a smaller lab, maybe a small research group, and you may not have all the computational resources or funding to have that kind of setup done for you. And that's where Kavatica could be helpful. And if you just like yesterday, David was sharing how to open an account on Kavatica and request for the $100 worth of pilot funds. That's where you could get started. Yes, Bijon, it is possible to get proprietary data. For example, that's what David was talking about. David, do you want to describe to Bijon how to get private data on Kavatica, control data on Kavatica?
Sure, yeah. So there's kind of different levels of privacy. So the demo that I'm going to do tomorrow is an RNA-seq demonstration, and it's going to be entirely based on open access data. So anyone can log in to the portal and access these files. So Kids First supports access, and I think I showed it to him on my talk yesterday. I had GText where I showed there's control files and then there's open files. And a lot of it depends on the content of the files and the ability for folks who might be misusing or not following the data agreement if they try to, the potential for re-identifying participants. And so things like raw alignment files are usually typically controlled, whereas these highly processed gene expression tables are open access because anyone can access them. So I'll go a little bit more into detail on tomorrow when I show the Kids First portal, but both of those file types exist on the Kids First portal. And so you'll be able to, tomorrow we're just going to move some open access files, but there is a way to apply for access through NIH dbGaP to get access to the private data files, so especially if you're interested in genomics. I saw people talking about, variant discovery and GWAS, and so for those types of analyses, you would need the controlled tier access. And so I'll talk a little bit about, how do you identify the access level of files? How do we apply? And then once you're approved, how are you able to move them? So in short, yes, it is possible to get, controlled tier data. And then even beyond that, the most private of data is maybe bringing your own data. And so if you have your own 50 samples, from a research collaboration that have been sitting in, and it would be great if I could combine those 50 samples with these 100 samples on the Kids First portal and get 150, and that would be even better. And so there are ways to do that as well on Kavatica. So if you have a cloud storage already, you can plug in AWS and access those files on your storage in your Kavatica environment. There's also opportunities to upload the data as well. So lots of different ways to connect.
Absolutely. So yeah.
Cynthia, I had one question.
Yes, please. Go ahead.
So from my experience trying to work with some data on Kavatica, one of the issues of which I encountered was like, if I'm trying to use the files halfway through, and then I want to process it further down in a different way, I had issues trying to understand how these, suppose I'm working with the BAM files, individual sample BAM files, and I want to work further downstream, so I should know the genome, reference genome, which was used early on, and things like that, and had some issues figuring that out, because there was some kind of a discrepancy somewhere running the tool downstream. So who would give that information? Is that information available somewhere for those files? How do we access that is a question.
Yeah, that's a great question. Yeah, so we'll show that tomorrow as well. So one of the inputs that we need for our RNA-seq workflow is the GTF, which has the a listing of the genes. And so I'll show how we can find that in the workflow, and then I'll show where we can find the exact file on Kavatica to plug it into our tooling. So I'll demonstrate that as well.
Yeah, thank you.
Mm-hmm.
All right, that takes me to like just the tools question. I was going to ask, how comprehensive is the toolkits for each of the data modalities that probably somebody's interested in working with? For example, for the transcriptomics, what are the toolkits that exist for like the analysis on Kavatica? I know we are not there yet, but it would be good because again, I have started exploring Galaxy, and I know they have like a library of tools that make things really simple. Is that the same thing that happens on Kavatica? Or there's a command line terminal, and again, the ability to like use inbuilt tools that exist on the platform?
Both, yes. So Kavatica has a library of these pre-built apps, which you could use for different data modalities that you may be interested in. And for each of these apps, Kavatica has a lot of documentation that they've written on what data to use to perform, to help that app perform efficiently, and what it is compatible with, and the kind of resulting visualization it will create, and the kind of they've also sometimes developed like public projects where just for demonstration purposes, there's like an entire end-to-end analysis that has been performed using one of the pre-existing Kavatica apps. So you could look at the existing public projects to see how to perform an end-to-end analysis, and you could also browse through the existing library of Kavatica apps and find an app that is suitable for the data modality you may be interested in, and just get your hands dirty with trying to get that app to work. Because you could start with something as simple as a plug and play, and then we can make it as complex as you may need it to be, with your own data that you could upload, or you could use a data repository where you may have access to some controlled data, or some public data that you may want to use through that pipeline and analyze. And yes, so the cost estimates on Kavatica, yes. Which is why, David, do you want to introduce everybody again to the concept of the pilot funds that you were talking about yesterday? Which is why
Sure.
it would be great if you could all get your Kavatica account set up before tomorrow.
Absolutely. Yeah. So some of our so this is kind of the tricky one of the tricky parts of working with the cloud is how much will it cost? And so especially if you're trying to scale and run 100 samples, 1,000 samples, 10,000 samples, how much is it going to cost? And so it's hard to predict something before you have done it, which is another tricky part. And so you can't really, there's not a great tool to just throw in your workflow, and then the tool will think about it, and then it will spit out a cost, 10 cents, $1, $3. And so the best way that we have is to run the workflow, on small numbers of samples and get a prediction, and then use that to scale up. So as far as our apps in Kavatica, some of our tools that we have developed, we've run, for our Kids First data, we've run it maybe 26,000 times, our alignment workflow. And so we have a really good sense of how much that costs. Whereas others are, maybe we don't have that same level of depth. So I can talk a little bit about cost estimates for some of the larger workflows and some of the standard Kids First workflows. And I can also show, this DEC2 analysis, which is going to cost probably less than $1. It'll run in, hopefully about an hour. So I'm just trying to give you a sense of scale here. But yes, we'll talk a little bit more about costs as well. I think especially highlighting again, as Sangeeta brought up, the opportunity to receive the $100 pilot funds, because this is where, it can go, $100 can get you quite a bit far on Kavatica, especially if you're, getting started, you're testing the water, maybe you're bringing a tool from your local server, you're experimenting with the different features. And so it can really get you a sense of, oh, this is what this is all about. This is what I can do here. And then you can, make that own personal decision to say, okay, I understand what it is and I'm deciding this isn't for me, or I understand what it is and I want to go further. How do I go further? And we can talk about opportunities through Kids First and through CFDE to receive additional cloud credit support, beyond that $100 if you choose to kind of continue on the platform.
Absolutely. So yes, please go ahead and set up your Kavatica accounts and send that email to support so that you can have your pilot funds allocated to you. If we don't have more questions, we are ready to wrap up.
I think so. Well, thanks for a great session today. Thanks for the very active chat. It was great to have engagement. Sometimes with these Zoom calls, it can be hard to to feel connected. But I appreciate all the interaction, the questions, and the participation today.